Good morning, my name is Ning Xiao, a PhD student in the Department of Chemical Engineering and Biotechnology at the University of Cambridge. Today, I'm going to talk about using a photosynthetic bacterium as a biocatalyst for hydrogen production. Hydrogen, a sustainable green energy, can be generated as a side product of nitrogen fixation process by purple non-sulfur bacteria in anaerobic conditions. From this simplified hydrogen production mechanism diagram, we can see the following 2 points. From the energy balance, nitrogen will be absorbed and converted into ATP to provide energy for bacterial growth as well as hydrogen production. And from the material balance, organic substrate will be consumed to provide carbon source for cell growth as well as electron source for hydrogen production. Therefore, both hydrogen production and bacterial growth will consume the energy and organic substrate. Quite often, we can use organic wastes, such as glycerol from biodiesel process, as organic substrate. A common soil and water bacterium called Rhodopseudomonas plastrace was selected in our project. It is widely applied in wastewater treatment industry, metabolically diverse, and it would not produce oxygen during the photosynthesis. This is particularly important for the safety of hydrogen production and storage. Here is a schematic diagram of the experimental setup. A culture bottle sitting on the top of hot plate magnetic stereo with the light provided by incandescent light bulbs. This type of light bulb was selected because it covers all the absorbance spectrum of the bacteria. The light intensity is about 174 watts per meter squared. Temperature is kept around 25 degrees C. Glycerol and nitrogen gas are used as carbon and nitrogen sources. I have prepared a small video to illustrate the experimental review. The culture bottle with specific PTFE bottle cap was used for this project. The bottle cap comes with 2 ports, a liquid port and a gas port. A temperature controller was used together with air conditioner in the lab to keep the temperature constant. One end of the temperature controller is connected to the reference bottle, and the other end is connected to the hot plate. The magnetic stereo together with the magnetic flea was used to provide mixing inside the bottle. Traditionally, people use single-stage hydrogen production method, so you grow the bacteria with glycerol and nitrogen gas and harvest hydrogen gas at the same time. The red dot is the cell concentration and the blue square is the cumulative hydrogen volume at 25 degrees C atmospheric pressure. From the results, we can see that our plastrace is able to grow on and convert glycerol into hydrogen. The specific growth rate of the bacteria is about 0.0157 per hour. The advantage of this method is that it produces hydrogen with high purity, always above 90%. But the disadvantage of it is the low yield, just around 10% in this case. Why the yield is so low? What are the energy and material used to? If we take a close look about the mechanism, we can see that the bacteria develop a nitrogen fixation process to absorb the atmospheric nitrogen gas and form ammonia for protein synthesis. Hydrogen is produced just as a side product. From the Haber-Bosch ammonia production process, we can see that it will take high temperature and pressure with a catalyst to convert hydrogen and nitrogen gases from ammonia, which means most of the energy and material are contributed to cell growth in this method. Ideally, we don't want that to happen. If we remove all the nitrogen sources, such as nitrogen gas in this case, all the energy and material is only used to produce hydrogen, and bacteria will perform as a live enzyme for the continuous hydrogen production. How do we achieve this hypothesis in the lab? Well, instead of single-stage cultivation, now we can divide the whole process into two stages: the cell cultivation and the non-growing hydrogen production. In the first stage, we simply grow the bacteria with glycerol and nitrogen gas as we did before. Once the bacteria reach a stationary phase, we harvest them, centrifuge them, and remove the nitrogen source, and recultivate them with glycerol only in the second stage. Here is a result of the hydrogen production and cell concentration at the second stage. Again, the red dot represents to the cell concentration, and the blue dot represents to the cumulative hydrogen volume. At the beginning, the bacteria still grew a little bit due to the nitrogen quota left from the previous stage. Towards the end, the hydrogen production stopped due to running out of glycerol. If we tidied up the results, we can see a constant cell concentration and a linear relationship between the cumulative hydrogen volume and cultivation time. From the plot, the average cell concentration was calculated as 0.405 gram per liter, and the bacterial growth was controlled within 5%. The constant hydrogen production rate was calculated as 4.75 ml per gram of dry biomass per hour. Again, the hydrogen purity was always above 90%, but more importantly, the yield of the hydrogen from glycerol was then increased from 10% to around 86%. Which one is better? The traditional single-stage method or the innovative two-stage method? Considering the feasibility in industry, the concentrated non-growing cells can be immobilized to give continuous high hydrogen production. Compared to the single-stage batch cultivation with low concentration, the two-stage hydrogen production method is more recommended. A possible example of biohydrogen industry plant would be like this. If the nutrients and solar power are supplied for the cell cultivation, the biomass can then be harvested for the second stage at the non-growing hydrogen production process. Afterwards, hydrogen can be collected and transferred to a hydrogen fuel cell. Potable water and electricity can be produced at the end products. I have also prepared another small video for demonstration. During the non-growing hydrogen production stage, the hydrogen was collected through the replacement of water inside the burettes on a daily basis. A syringe and stopcock was used to extract the gas sample out. Normally, the gas sample was then tested by a gas chromatographer for its gas composition. But in this video, it was injected directly into a hydrocar module to test the purity of the hydrogen gas produced. About 20 ml of hydrogen gas was then injected into this car module, and the car can only consume very pure hydrogen based on the instruction. Now, let's test the hydrogen purity by switching it on. The car started to move. The hydrogen produced was converted into electricity by the blue hydrogen fuel cell and used to drive the car module. For 20 ml of hydrogen gas, it will support the car module to run 20 minutes. Obviously, I'm not going to show the car module running for that long, so let's go back to the presentation. How much hydrogen we can produce, and what we can do about it in our daily life. For example, for 1 gram of dry bacteria with 1.8 ml of glycerol, it will produce about 114 ml of hydrogen gas at atmospheric pressure and 25 degrees C. And the 114 ml of hydrogen gas is equivalent to 1,440 kilojoules of energy. If the hydrogen fuel cell has 100% conversion efficiency, then the energy for this amount of hydrogen gas could supply the following household appliances for the following period of time. For example, it can support a double-door fridge for 45 minutes, or the 2-liter kettle for 30 minutes, or the toaster for 20 minutes, or the washing machine for 15 minutes. Or if you want to heat up the food, and you can use the microwave for 20 minutes, or simply watch TV for 160 minutes. Or when you feel hot, you can run the air conditioner for 25 minutes. So that's how we started our project with my supervisor and collaborators. Apart from what we are doing now, the optimization of hydrogen production, the light impact on cell growth as well as hydrogen production, the hydrogen fuel cell, we are planning to do a little bit more. We want to investigate on different methods and techniques about cell immobilization, such as alginate beads or hydrogel biofilms. In this case, we can grow the bacteria, immobilize them, and use them as a biocatalyst for hydrogen production in industry. We know that bacteria can utilize a lot more other than the glycerol. So we want to take a close look into many other waste treatment for different scenarios. At this point, I want to say my special thanks to the Cambridge-CAS Joint Research Program for the generous funding provided. Again, our collaborators over the world, Professor Fushen Liu in China, Madalena Lemos in Portugal, Dr. Cameron Younes, Dr. Vasilius Vasilides, and Dongda Zhang in our department. Last but not least, I want to thank the supporting staff in our lab and our department. Here is a list of the references in this presentation. If you have any inquiries and questions about this presentation, please contact me through the emails, and thank you for your time and patience.